So in my previous video, I have told you that how erythropoietin stimulates the production of pro-erythroblast from hematopoietic stem cells and in turn erythroblast from pro-erythroblast. This I have explained in my first video about blood physiology, that how pro-erythroblast differentiate to form the mature erythroblast. Now, now I have also told you in the previous video that how this causes the stimulation of this. So this is a complete, but now we will study that how much time it will take. It will the maximum production of erythropoietin will be in 24 hours. Means that if there is low oxygen or low blood volume, our kidneys start producing erythropoietin and it reaches to its maximum limit within 24 hours. So erythropoietin reaches to its maximum limit in 24 hours, and obviously when erythropoietin increases, this causes the increase in stimulation of erythroblast from pro-erythroblast. So RBCs concentration increases in our body, but this EPO which will cause RBC production after 5 days. Why after 5 days? Because there are some steps which, for example, the production of pro-erythroblast from hematopoietic stem cells and then erythroblast from pro-erythroblast. So this takes a period of 5 to 6 days, after which our RBCs start increasing inside our blood, and this will be the response of hypoxia in our body. This will be the means if there is any renal hypoxia or non-renal hypoxia. Now, obviously, erythropoietin is the main element that stimulates the production of erythroblast, but there are some other factors that are also responsible for stimulation. If there is much iron, if there is sufficient iron in our body, then there will be a huge increase in RBCs. And the other two factors is the B12 and B9. Folic acid we call it B9, and this is B12, and both of these are used for the final maturation of our RBCs. So both of these are also important. B12 is required in our daily amount in 1 to 3 micrograms, and this is stored in liver. So main factor is erythropoietin. After the production of erythropoietin, if there is much iron and sufficient B12 and B9, there will be a huge production of RBCs which can respond to the hypoxia condition. Now, how both of these vitamins B12 and B9 stimulate? Both these hormones cause the final maturation of RBCs. How causes the final maturation of RBCs? Because both of these are used for the production of thymidine triphosphate. When there is TTP increase in our blood, when there is TTP increase, this causes the DNA stimulation to generate the erythropoietin production. So this DNA will cause the final maturation of RBCs. So both these are factors, means that for RBCs production we need erythropoietin, then we need iron, then we need both of these hormones. These all are important for the RBCs production inside our body. So we have discussed from here up to here. Now we will study if there is absence of B12 or B9. If we are low in both of these vitamins, then the RBCs can not be well differentiated, cannot be, their final maturation will be not done. If there is not final maturation, the RBCs will be macrocytes. They will be large. This membrane will be fragile, and these are large, oval shaped will be formed, and nose with spherical, almost oval to spherical shape. And when these passes through the capillaries, they usually die. Usually we have RBC's lifetime is 120 days, but if they are biconcave. But now if they are macrocytes in the absence of B12 and B9, they will be oval in shape, and their lifespan will decrease from 1/2 to 1/3, means 60 to 30 days. So these are the factors, and in the absence of these factors, this can happen. Now the main condition that can happen in the absence of both of these hormones is the pernicious anemia. We know what is anemia. Anemia means low hemoglobin inside our red blood cells. So when there will be low hemoglobin and pernicious means nutritious. Pernicious can like to the word nutritious. Nutritious means that something we're taking in diet. If we're not taking B12 and B9 in our diet, then we will be suffering to the pernicious anemia. How this pernicious anemia happen? Whenever we have atrophic gastric mucosa, whenever we have gastric mucosa, means that atrophic gastric mucosa means that parietal cells in our stomach are atrophic. Whenever these cells are atrophic, there will be no intrinsic factor. What is this intrinsic factor? This is the protein that is released by the parietal cells of gastric mucosa. Now this intrinsic factor binds with B12 in our diet, present in our diet. Now when both of these are joined together and causes the absorption of B12 in the intestine. Where in the intestine? In the small intestine, through pinocytosis. So parietal cells are responsible for the production of intrinsic factor, which in turn combines with the B12 and in turn causes the absorption of B12 inside the ileum, in the brush-like layer of the ileum, through pinocytosis. Now when there is absorption of B12, it is then stored in the liver, and whenever we need RBC's production, liver, a small amount of stored B12 is moves from the liver towards the bone marrow, and bone marrow causes the production of RBCs from the hematopoietic stem cells. So this was a whole discussion about the erythropoietin rule. Also we have learned that how other factors stimulates the production of RBCs. So this was the whole diagram. Once again, I'm repeating this, that parietal cells in the stomach, in the gastric mucosa, produces intrinsic factor, which in turn combines with the B12 and causes the absorption in the small intestine, in ileum, through the pinocytosis, and then B12 is stored in the liver, and whenever we need RBCs production, B12 moves from here to the bone marrow to cause the RBCs production. So but liver stores a huge amount of B12, about 10,000 times we need daily. So if we are deficient in B12, then its deficiency cannot be recognized early, because we have sufficient amount here. Its deficiency will be felt after receiving